putting together this wonderful seminar series. I've enjoyed a lot of them, so I'm honored, and it's really my pleasure to be presenting to you guys today. So I'll be talking a little bit about models of resistance in structured populations, and I'll present sort of 3 example cases of how modeling can shed light on therapeutic resistance in cancers of different kinds. But before delving into this, I sort of thought I wanted to share a few words on what the current state of the field is in cancer research. And so this is an excerpt taken from a short story from one of my favorite authors, Edgar Allan Poe. And it reads, he impaired his vision by holding the object too close. He might see perhaps 1 or 2 points with unusual clearness, but in doing so, he necessarily lost sight of the matter as a whole. And I think this is a really apt characterization of the field today, in which most of the cancer researchers really focus on a specific gene, a specific protein, a pathway, without taking this broader perspective and considering how their piece sort of fits into the broader puzzle that is cancer. So with that in mind, I sort of want to reframe our thinking from cancer as a disease of the genes to now cancer as a disease of evolution. So what's the problem? So this is perhaps the most overused example of treatment failure that's shown in all clinical oncology talks. So this patient here presented with an aggressive form of melanoma, which is a skin cancer, and was treated with this RAF inhibitor, a chemotherapy drug. And 15 weeks post-therapy, we see in the middle panel here that he's all clear. His cancer is declared cured, he's sent home. But this last panel is taken just 8 weeks after the second picture. So we see that his cancer has returned, it's worse than ever, and the main reason for this is therapeutic resistance. And so as a mathematician, as a theoretical biologist, let's sort of take a theoretical modeling approach and see what insights we might be able to gather about this problem. So this is the basis for the modeling framework we'll use. It's called the G-function framework, and it's really just this set of coupled differential equations. So the first equation, dx dt, captures the population or the ecological dynamics of our cancer cells. So x is the current population size, and g is this g function, this fitness generating function, which captures the per capita growth rate of this population as a function of v, which is its strategy, so something like drug resistance, and x, the population size. And then the trait or evolutionary dynamics are then just derived from Fisher's fundamental theorem as the product of evolvability, K, and the selection gradient, dg dv. So to see this a little bit more intuitively, we can plot this G function on what's called an adaptive landscape, where we're plotting the fitness as a function of the trait value here. So if you imagine your cancer cell at this yellow circle on this landscape, you're going to want to scale the adaptive landscape to some peak. And so here you'd want to scale up this peak to this green star at A. And the way you do that is dependent on 2 things. So first, again, evolvability, which is the capacity for a cell to generate heritable variation that can be acted upon by natural selection. So you can think of this as a step size. And dg/dv, or the selection gradient, which is really the slope of this landscape. So the steeper your slope is, the larger your step size is, the faster you're going to evolve. And in previous work, we theoretically examined the importance of this evolvability parameter in a variety of evolutionary contexts. And broadly, we noticed that evolvability is really a double-edged sword. So in stressful environments, the benefits of having a high evolvability outweigh the costs. But in more stable environments, the opposite is true, because you can imagine if you're at a peak on your adaptive landscape and you're generating all this heritable variation, by definition, a lot of that's going to be unfit progeny, and you're going to get some sort of mutational meltdown. So a natural next question for us to ask is, if there's something called facultative evolvability, right? So what if cells could modulate their evolvability in response to environmental conditions? So namely, if you increase the evolvability during bad times when you're really stressed, and then decrease it during good times. So how might this happen? We know in bacteria, there's something called stress-induced mutagenesis. So when bacteria are stressed by nutrients or a drug or something, they tend to upregulate their sort of level of mutagenesis. Does this happen in cancer? Turns out that it does in what's called these polyaneuploid cancer cells. So this will sort of be our first vignette, our first application case. So what are these cells? So polyaneuploid is really composed of two words, polyploid and aneuploid. So polyploid referring to a duplication in a genome, and aneuploid referring to a genome with an abnormal number of chromosomes, which a lot of cancer cells just normally have. So then polyaneuploid refers to a whole genome duplication of this aneuploid genome. And this PAC state is really formed when aneuploid, or otherwise known as 2N plus cells, bypass mitosis or cytokinesis or both, and undergo this whole genome doubling. So the main characteristics here are that the cell exits the cell cycle after S phase and pauses division. So this allows the cell to avoid DNA damage or apoptosis, as might be sort of induced by therapy. And you also get this increase in genomic content due to this polyploidization program. So they get really big, right? And we can see this in even the panels here, where these really large cells that I'm showing here are these PACs. So they're visually much larger than the normal cells, and you can detect them just like that under the microscope. And what makes them really dangerous is that we think that there's some sort of condition-dependent formation. So they occur at pretty low levels under baseline, so around 2 to 5% of the cancer cell population might be in the state under normal conditions, but they become incredibly common when the population is under stress. So they can be up to 80, 90% of the population under extreme therapy. So we hypothesize that this, again, the PAC state forms facultatively. So it can be modulated up, increasing the rate of evolution, or down, decreasing the rate of evolution as a strategy contingent upon the cancer cell's condition. So in thinking about this, we really have to stress and keep in mind that the PAC state exists as a cell state in the life history, the life cycle of a cancer cell. So it's not a separate cell type. So you can imagine, for example, a butterfly and a caterpillar. If you didn't know any better, you might think they're two separate species, and you might model them as such. But ultimately, that would be incorrect, especially when we're thinking about evolutionary components, because these two states are really tied together. What happens when you're a butterfly impacts the caterpillar state and vice versa. And many things that we treat as different cell types, especially in cancer, implicitly may actually be different cell types. For example, sensitive and resistant cells. So it's really important to keep this in mind. And what this means for modeling is that we need to then come up with a unifying fitness function that can track the evolution of a trait such as drug resistance across these 2N plus and PAC states. So to do that, we've sort of developed this method basically that you can start from this life cycle graph or this set of equations. You can throw all that into a matrix, and then you can take the spectral radius or bound as this notion of the G function, this per capita growth rate, and then you can go back to the equations I showed earlier and then continue with your eco-evolutionary framework just like that. So here's just a basic model of the PAC ecology and demography. So the X1, the first equation, represents the dynamics of the cells in the 2N state. The X2 represents the dynamics of the cells in the PAC state. So very basic equations. All we have is we have some growth in the X1 state that's not in the X2 state, right? Because these cells don't divide. You have background transitions between both the states. You have drug-induced death in the 2N state that's not there in the PAC state. And then again, you have this facultative formation of the PAC state that depends on the cell death term. So we can obviously make this model more complex if needed, but for now, this is all that we need. So with that, we can then come up with hypotheses and then simulate these hypotheses and see what happens. So we have 3 hypotheses here. And in all these plots, the blue lines are going to represent the cells in the 2N state. The red lines are going to be those in the PAC state. And then the black lines in the bottom panel for each of these sets are going to be the evolutionary dynamics. The green regions are the regions of therapy, and the white regions are when no therapy is administered. So standard tumor heterogeneity, this is what is commonly used in the cancer literature. And all this means is that there's no PAC state. So what we see is if you give a low dose of therapy on the left-hand side here, they do just fine. They're able to undergo evolutionary rescue. They're able to evolve a resistance strategy fast enough to remain in the population. But when we give them a really high dose of therapy, the population crashes and goes extinct. It can't evolve resistance fast enough. Then we look at non-proliferation. So this is sort of like a dormancy or hibernation strategy. So all that means is that when therapy is administered, these cells just switch into the PAC state. There's no evolution of resistance going on. They're sort of just hunkering down. And then once therapy is removed, they can, again, switch back to the 2N state and continue proliferating. Our last hypothesis was this evolutionary triage. This is the one we really thought was right. in which we notice that there's a transition to the packed state again upon the induction of therapy. But because that packed state not only serves as a refuge from therapy for cells, but it also contributes to the evolvability of them, we notice this, again, greater generation of heritable variation, faster evolution. And so as we can see, over time, as the population gains resistance, the frequency of packs decreases, right? Again, because of that trade-off between cell division and resistance. We also consider other things, life history inspired sort of therapies and stuff. I won't present that here, but obviously what we do now is we have these models, then we have to go back and look at the experiments, right? What are the experiments saying? And as I'm sure all of you know, it's usually wrong, right? So what we see is that under extreme stress, there is a drastic transition of cells from the 2N state to the non-proliferative packed state. And after a delay, cells in the packed state begin depolyploidizing into 2N plus progeny. So that's generally okay. But what's really critical is that those progeny themselves immediately display high levels of resistance to the stressor. So unlike we would expect under evolutionary triage, classical Darwinian evolution with Mendelian inheritance, we don't observe the death of mutant clones. So the evolutionary process doesn't require this creative destruction. It appears to solve this therapeutic problem without the mortality of less successful variants relative to more successful ones. So that means we need to come up with a new hypothesis. So we think that there might be some sort of self-genetic modification or reverse transcriptional inheritance going on. What does that mean? All it means is that changes that a cell undergoes during its lifetime, for example, within the packed state, are then written back into its DNA and passed on to its offspring, so that when the pack depolyploidizes, those cells then are resistant. And existing evolutionary models can't really account for this because, again, this does have a little bit of a Lamarckian nature of inheritance to it. So what we do is we then use or develop this sort of stochastic birth death switching and mutation process. And this is functionally just a Gillespie algorithm with this evolutionary component tacked on. So basically, from our system of equations, we can compute all the birth, death, and switching rates. We can choose the event type and type the event probabilistically, and then we carry out that event. And if it's a division event, then we carry out a mutation, right? We induce a mutation with some probability. And due to its greater genomic content, as mentioned before, the packed division events are going to generate a greater mutational breadth. And if that mutation confers a higher fitness, we'll replace the resident trait with this mutant trait. But if the drug kill term in this green section here, right, if that exceeds some threshold, some self-check modification threshold that we set, right, in this packed state, then what's going to happen is that evolution is going to happen behind the scenes. Right. So rather than the cell depolyploidizing directly into the 2N state, evolution is going to sort of happen behind the scenes until the cell in the packed state can sort of find a good enough solution. Right. So the mutant traits with the higher fitness will still replace that resident trait, but it's not going to show up in the population because that cell in the packed state is not going to divide. It's not going to depolyploidize. It's going to keep trying to figure out a solution. You're going to go through the exact same process until it finds a good enough solution, then it depolyploidizes, that progeny gets that resistance value, and then they can go on continuing to proliferate just like sort of evolutionary triage, right? So how does this actually look? So the results for evolutionary triage are on the left, and those for self-genetic modification are on the right. And sorry, it looks like I've switched around the colors here on accident. So the packs are actually going to be blue here, and the 2N cells are going to be red. But evolutionary triage, notice the same thing as before, right? We notice this rapid increase in packs upon the induction of therapy, and we see this gradual shift towards the 2N cells. But as you can see, evolutionary rescue here is insufficient in most of these cases, right? Most of these cases go extinct. But under self-genetic modification, we see this really clear delay here, right? Where basically all the populations, all the cells are in this packed state. There's a delay, and then once they found a good enough solution, the evolutionary strategy jumps up, and then everything goes back to normal, just like evolutionary triage. And this avoids extinction in all cases. So, you know, you might be asking then, okay, well, if that's such a good solution, why don't cells always do this? And one thing that we haven't really mentioned here yet is the impact of density dependence, right? Because again, there's this trade-off between cell division and resistance. So with that in mind, we sort of ran these in silico competition experiments in which we basically competed cell populations that evolve via evolutionary triage with cell populations that evolve via self-genetic modification. And we basically noticed three different regimes. So this first regime is the neutral regime. Again, it depends on which threshold we set for the self-genetic modification threshold. So which threshold we think is good enough for the Paxton depolyploidize. But here in this neutral gray regime, evolutionary triage and self-genetic modification are operating identically. So here, when we plot the extinctions, right, out of 100 different trials, they're operating identically. Both strategies do the same thing. But in this sort of sea green region in the middle, when the cells are somewhat stressed, we see that evolutionary triage really has an advantage, right? You have much less extinctions noticed here. Again, because of this density-dependent advantage. Here, cells are really concerned with outcompeting their neighbors, outcompeting the other population. And so this evolutionary triage has the advantage because cells in the 2N state can still continue proliferating, whereas under self-genetic modification, they can't. But when you're under this very stressed regime, this yellow regime, then you're not really too much concerned with what your neighbors are doing. You really just want to survive the therapy. So when the situation's like that, self-genetic modification really has the big advantage. So with that, that'll sort of be the first vignette. Here, I'll talk about a different application, similar flavor of modeling, but with neuroblastoma. So this is joint work, again, with Sophie Mullen and Stina Anderson at Lund University, who are the experimentalists here. And we work with neuroblastoma. So what is neuroblastoma? It's a pediatric cancer, brain cancer, and it has really variable clinical presentation, right? So we see anything from really high stage metastatic invasive cancers that are therapy resistant all the way to spontaneous regression, where the physician doesn't do anything and it just goes away on its own. So the mechanisms underlying the initiation, progression, emergence of resistance are really not understood here. But what we do know is that there seem to be two cellular phenotypes that underlie these responses. So the adrenergic, or ADRN, and the mesenchymal, or MES. So the ADRN cells are differentiated, they're drug resistant, they're more proliferative and less motile, whereas these MES cells are more drug resistant and less proliferative and more motile. So those are the key trade-offs to sort of keep in mind. And in the literature, there's really a lot of confusion as to what these cells are. So this is just an excerpt from like the first paragraph of a high impact factor journal article on neuroblastoma here. And as you can see, they call these cells phenotypes, cell states, and ADRN or MES type cells, all in the same paragraph, right? Written by the same people. So really the understanding of why these distinctions are important, much less so what they actually are, is sort of lost among most of the researchers here. There's another question too, as to whether ADRN cells can evolve at all. So that's also sort of a hotly debated topic here. So we take this sort of modeling first, experiment next approach. So we come up with 4 different hypotheses, sort of combining all the possibilities here. So our hypothesis 1 is if they're cell types and the ADRN cells don't evolve. Hypothesis 2 is if they're cell types and the ADRN cells do evolve, and then 3 or 4 cell types, again, without evolution and with evolution. So then we can construct some equations for these guys, we can run some simulations, and then we can then inform experiments, test those with the experiments, and then sort of decide what we think is going on. So again, very basic equations, cell types without evolution. So the first equation is for the ADRN cells. The second equation is for the MES cells. We just have basic growth, background death, and drug-induced death. Very easy. And we run some simulations here. As you might expect, in the regions when therapy is not administered, the ADRN cells have the big advantage here because, again, they're more proliferative, so they dominate the population. But during periods of therapy, the MES cells increase in frequency and number here because, again, the trade-off of them being resistant to therapy during this period is much more than the downside of them not being as proliferative. And critically, when we administer therapy again and again in this sort of intermittent fashion, you notice the exact same dynamics each time because there's no evolution of resistance. When we consider them as cell types with evolution, so then just adding in this evolutionary equation, again, from the G-function framework, we know similar dynamics. So here, our bottom panel, we've again introduced the evolution of resistance here. And the key thing to note are the dynamics upon repeated administrations here. So due to the evolution of resistance, the MES cells have less of an advantage during each subsequent administration. So they still increase in size, but there's less and less of an advantage there. We can then consider cell states without evolution. So now we have these transitions back and forth, again, both background and facultative. And this is very similar to our non-proliferation sort of hypothesis, basically just switching states, right? And during the periods of therapy, we notice this, again, stable dynamics in the MES state. They aren't changing at all. They just switch in. And dynamics upon repeat administration, again, as you'd expect, the exact same because there's no evolution of resistance. When you add in the evolution of resistance, again, throwing all this into a matrix because now this is a state-structured population, we have similar thing to what we observed with the PACs. So upon the induction of therapy, they shift to the MES state. As resistance evolves, they decrease in frequency. And again, with repeated administrations, less and less of the MES advantage. So with that, we can then come up with ways that we can falsify these hypotheses. So the first question we have is, do cells with the ADRN phenotype, do they evolve resistance? So we can determine that based on the dynamics upon repeated administrations, right? So if they're different, then yes, they do evolve resistance, right? Specifically, you'd expect the frequency of cells with the ADRN phenotype to increase and those with the MES phenotype to decrease with subsequent therapeutic results. If they don't, if they're basically the same each time, then they probably don't evolve resistance. And then our second part, are these phenotypes cell types or are they cell states? So if they're cell types, right, then we think we can determine that by the MES frequency and number during therapy. So if the MES is increasing, right, the whole time, they're probably types. But if they're cell states, then we expect the MES to increase, right? Upon the induction of therapy, we expect the MES state to increase, and then we expect it to remain stable or decrease, right? So with that in mind, we can then devise an experimental plan. So this is an experiment we're now currently running where we can culture these cells until confluence. We can measure their pre-therapy frequency and counts using various markers for these cells. We can treat them. We can measure the frequency and counts again. So this is where we distinguish between type and state. Then we can allow them to recover to pre-therapy conditions again, and then lather and secrete, do this again and again. And that's when we can really tease apart this evolutionary aspect of it. But who cares, right? So I mentioned at the beginning that people don't really even think about this, right? So why should we? So, well, there's a couple of reasons. One is, as we've already seen, there are different ecological and evolutionary dynamics. So if you're a physician and you're administering therapy to a patient with neuroblastoma, I think it would probably be useful to be able to predict or have a notion of how the therapy is going to go, where the evolutionary and population dynamics are going to go. And so without understanding if they're cell types or cell states, we won't really be able to do that effectively with any kind of mechanistic understanding, at least. The second aspect are these life history enlightened therapeutic strategies. So this is something that I briefly mentioned with the PACs. And so if they're cell types, for example, and it's really hard to target these MES cells because, again, they're really therapy resistant to whatever therapy we're giving them, we might be able to devise these life history enlightened measures. So what this means is that instead of targeting those cells directly, maybe we can try to target the transition. So we can prevent the cells from transitioning into or out of that state. That might be an effective way about going about treating these guys. But throughout both of these sort of vignettes, we've assumed that the states are discrete. We've assumed there are 2N cells, we've assumed they're PAC cells, we've assumed they're ADRN cells and MES cells. But not everything is discrete. So this very recent article came out in late 2022, showing that the ADRN and MES states really exist on this sort of trajectory, this sort of continuum, right? There are some transitional states that exist. So we might not be able to just deal with them as this binary switch. We know this exists all across cancer as well with the EMT transition, epithelial to mesenchymal transition. There are not just two phenotypes. There's really a continuum, right? There's really, and same thing with the macrophage polarization, right? With the immune cells, there's really a different roles that these cells play, right? It's not just a discrete switch. So we need a different mechanism to address these questions, a different modeling approach. So to deal with these continuous structured populations, we're going to use what are called these integral projection models, or IPMs. And these are based on the three central tenets of life history theory, which are birth, survival, reproduction. And functionally, right, at a bird's eye view, this is the same approach as the discrete state case. But instead of using our population projection matrix, we're going to use a kernel to capture these components. So this is the base sort of eco-demo model here. So this is just n of t plus 1 as a function of the drug resistance u and the current state z is given by this big kernel here, right? And we can decompose this further in the second equation. So the first term is the transition kernel. So that's given as a product of survival and then this growth kernel, right? So they survive, and then where do they go? Where are they sort of diffusing? So again, z is the current state, and z prime is the state at t plus 1. So here we're just assuming a normal distribution for this, but we can plug in whatever we want. And the same thing here. The second term here, this is called the fecundity kernel. So there's a birth rate dependent on the current state. And then we have this reproduction kernel, right? So where are those progeny going, right? And again, assuming a normal distribution here. So that's the base eco-demo model. And then again, we can derive this evolutionary model the same way we do with our G functions. So here the X bar for any of these guys is just the mean value of this in the population. Lambda is the per capita growth rate, which again is calculated as a spectral radius of that kernel operator. And then this SUZZ is the sensitivity function of the fitness function, sorry, to perturbations in this kernel operator. And we can basically just write this out with this basic application of this chain rule. So now we have the ecological model, the demography model, we have the evolutionary model derived just as before, and then obviously we implement this numerically, in which it functionally becomes very similar to our matrix approach. We just have this very large matrix now functionally, right? Because we discretize it, we lay this mesh over it. And that mesh size, that's an important factor to keep in mind, how fine do you want it. So obviously there's this trade-off between accuracy and computational efficiency, and sort of heuristically or practically when you do that, you just want to make sure that your whatever size you use is not too coarse that you end up getting different results. So the stability is not there, which is just verified numerically. And then because we have these Gaussian functional forms, these normal distributions, you have to keep in mind that you have to sort of carefully enforce your boundary conditions. So here we just sort of push them to the ends, right? To prevent them from just getting evicted and ignored in our accounts. We'll just set this defined boundary, right, for the ADRN or MES states, for example. So with that, just the last aspect of this is going to be a little bit more general. So it's just general stress responses in bacterial and cancer populations, more so just to illustrate how this approach will actually look. So this is the no evolution case. So if there's no evolution at all, on the left panel here, we're going to look at the cell state. So this has been bounded by 0 and 1, and the higher states are going to be more resistant, but less reproductive. They have that sort of birth resistance trade-off again. The second population is going to be our population size, and the third is going to be the drug resistance. And we've induced therapy to go whenever the cells hit this million cell population threshold. So what happens is that before therapy, we see cells that are moving down to lower cell states, which makes sense because the lower cell states have this higher reproduction rate. Once therapy is administered, again, remember there's no evolutionary resistance, so these cells just shoot up to higher cell states. Unfortunately, they haven't really recovered here by the end of the simulation. They've sort of gone extinct. And you can visualize sort of the distribution here of the population density across the cell states and this sort of thing. So before, again, we're moving towards lower cell states. Therapy is going to hit now, so we move towards the higher cell states, and we stay there because, again, there's no evolutionary resistance, right? So then we can look at a control case where now we've allowed the cells to evolve some sort of resistance. So the key thing, again, before therapy, basically the same thing. You're moving towards lower cell states. But now, because of this evolutionary resistance, although you get this quick switch, right, to higher cell states, because that adaptation phase is generally faster than the drug resistance phase, after that happens, because of this evolutionary resistance, you start moving back because, again, of this trade-off, right, birth and resistance trade-off. So now you decrease your cell state back again, and the population undergoes evolutionary rescue, it's perfectly fine here. And so you can see this again paralleled in the state distribution dynamics. So therapy is going to hit and you move over, right? And then as cells again start gaining resistance, the population is going to slowly move back towards close to pre-therapy sort of frequencies, distributions. So then we looked at a lot of things. I won't go through all of them here. We've looked at various different forms of sort of facultative stress-induced responses, like facultative plasticity, adaptive mutagenesis. Here I show directed mutagenesis. And in all of these cases, you notice this higher minimum, so the population doesn't get affected as much by this initial therapy, and you notice a shorter time to progression, right? Which is just the time to when it hits the threshold again, and time to failure, which is when the therapy starts failing, than the control case, as you might expect, right? And so again, here with directed mutagenesis, you're going to notice the shift back to lower states, and you're going to notice just a small shift, right? Because of this really quick evolutionary resistance, because of this facultative evolvability aspect of it. And then so far, we've just looked at sort of continuous therapy. What happens if you do adaptive therapy or intermittent therapy? We looked at intermittent as well. And what we noticed generally are that introducing drug holidays leads to higher nadirs, as you might expect, obviously, a lower time to progression, because again, you're not diminishing the population to as low of a population size as possible, but you do notice higher time to failure. So what that means is that if you have several drugs and you're aiming for multi-drug control, which is usually what happens in adaptive therapy, that's usually your goal, it's better to introduce these drug holidays, right? And so again, the state distribution here, which look a little funky because now you're moving back and forth sort of rapidly, but generally you're still going to be trending towards the higher cell states for now because of the evolution of resistance. So with that, just a brief summary here. So genes are important, yes, of course. No one's saying that genes aren't important, that sort of taking a gene level, protein level, a pathway level perspective is not important. But it's also critical to recognize that there are other aspects that we need to consider. This really common quote here, famous quote, the evolutionary play takes place in the ecological theater, with sort of a little of my added touch here with demographic actors, because it's really important to consider these different cell states. So not just evolution, but sort of adaptation is critical to take into account, plasticity. And then again, assumptions are critical in general, but here specifically thinking about cell types and cell states, because obviously those are not the same thing. So with that, I'd like to acknowledge my team here. So Emma Hammerland is currently my host at Lund University, where I'm a guest researcher. Joel Brown is my PhD advisor back in Moffitt. Ken Pienta, Bob Austin, Sarah Amend are other collaborators on the PAC project. Sophie Moll and Steen Anderson, who have been critical in our neuroblastoma sort of investigations. And then I just want to thank my funding sources. So with that, happy to take any questions if you have them.